Hi everyone, my name is Shijing Feng, and I'm very glad to have the chance to talk about something about the basics in downstream analysis of single-cell RNA-seq data analysis. So, in this part, we're going to talk about some important steps in single-cell RNA-seq data analysis, which starts from data filtering to normalization, dimensionality reduction, marker gene detection, cell annotation, as well as a brief introduction to the enrichment analysis. So in the next week, we are going to have a lecture about the advanced analysis, and all that these analysis will show based on the results from these steps. So after cell calling, what we got is the matrix data, which is the raw data counts. And what we do is to first filtering out unwanted cells and do normalization, which is very essential for downstream analysis. And after we got the normalized data, what we will do is to do the dimensionality reduction as well as the clustering. And in many cases, we are dealing with multiple samples from multiple batches, and what we do is to considering the battery effect. But today we are not going to talk much about the battery effect correction, because we have a lecture later talking about how we can do the data integration and how to remove the battery effect. Also, imputation is not an essential process. However, if there are severe RNA contamination in your data, maybe take a look at one of these listed softwares here. So before we start, I want to introduce two popular toolkits. Both are widely used in single-cell data analysis. One is ScanPy. It is written in Python and have a very high capacity to dealing with very large data sets. And if you have the requirements to integrate a great number of samples for the analysis, or you are more familiar with Python, so ScanPy is a good choice. In the other hand, scRNA is developed in R, and even though there will be some problem when dealing with extremely large data sets, R is very good for data visualization, and a lot of analytical tools for single cells are developed in R. So still there are a lot of people try to use scRNA as well for the analysis of single cell data. Even though the two pipelines are not exactly the same, so the main concept of every step is very similar. So today I will not focus on the codes, but on the concepts of why we do these steps and how we could choose a good parameter for the analysis. And if you are interested in practicing the data, so both of the websites I listed here have very detailed tutorials for you to know how to do the analysis, and maybe you can try to play with it. And today, most of our examples are from scRNA and by using this test data. So let's go to the first part, which is cell filtering. In cell calling, we have filtered out some empty cells. However, there are still unflavored cells that you're not going to use for a later step. So we do extra data filtering here before we start. According to the parameters of the number of RNA molecules in the cell and the molecular genes expressed in the cell, as well as the percentage of mitochondrial genes expressed in every cell, we shall filter out some of the cells that we are not going to use. So basically, cells with very high RNA content tend to be the doublets or multiplets, and doublets and multiplets are artifactual libraries generated from two or more cells. So if it is droplet-based technologies like 10x, the two cells will tend to flow into one droplet and forms a doublet. Or if you are using the microchip-based technology like Singleron, two cells may fall into one hole to form the doublets. And the percentage of the doublet is correlated with the concentration of the cell suspension you loaded. And even though when you follows the industry's instruction, so we still see some doublets in our data. So it is a very common phenomenon. In the other hand, cells with very low transcripts tend to be noise or cell debris. So by filtering, by setting the threshold for uncount RNA and feature RNA, we are filtering out these types of unwanted cells. So on the other hand, we will filter out these cells with very high content of mitochondrial gene expression. So these cells tend to be noise, and these cells are sometimes hard to annotate because the upregulated genes are all from almost from empty genes. However, so the percentage of mitochondrial genes is relevant to the tissue type. Some of the tissues may have very higher percentage of the mitochondrial gene expression. For example, the heart tissue, we usually see the percentage of empty gene in heart tissue is around 50% or more. And it is because that the heart needs a lot of energy to keep pumping. So they will have like higher percentage of empty genes. So you have to check the tissue type when you are doing the analysis. So I would suggest to check your these type of parameters by visualizing into dividing plots to see the distribution in case that you filtered out too many cells for the downstream analysis. So after that, we're going to do the normalization for raw count data. So, so normalization is an essential part because the sequencing parts of different cells can be very different due to the technical reasons. And sometimes when you integrate different samples together, the sequencing coverage of different samples or experiments from different batches can be very different. So normalization have to be done here, especially when it is important because when you are doing the differential expression analysis, what we want is that all the DEGs are from a real biological difference, but not the technical variations. So, so before doing these types of analysis, we have to do the normalization. However, there were a lot of normalization methods. So which one to choose? So it is relevant to the technology you choose to use. So for example, for full length data like the RNA-seq or SMART-seq2, so here all the reads are used for quantification. And in this case, we are not only normalizing the sequencing depth, but also the exon or gene length, because longer transcripts tend to have more reads mapped to it. So here I would suggest you to use FPKM or TPM for the normalization. So for example, FPKM is calculated by the total exon fragments of that sample or that cell in SMART-seq2, and divided by the mapped reads of that sample or cell in million, as well as the exon length in kilobase. And after that, we got the normalized data. However, if it is 3' or 5' and tagged data with barcoding, like we usually do with 10x genomics or single JAK-scope, it is a little bit different because all the reads are captured by the bead and pulled together for sequencing. And the NGS produced reads with very limited length, for example, 150 BP. And in this case, only the reads with cell barcodes, which is close to 3' end or 5' end, are used for quantification. So in this case, the exon length or gene length are not important. So, and therefore, the different normalization tools shall be used. Here, I would suggest you to use log normalization or CPM. So both of the methods are done for every individual cells by divided by the total UMI count, which is the transcripts of these cells, and it is related to the sequencing depth. And the difference of the normalized count and CPM is that we will do an extra log transformation for normalized count. And we will see that the log normalized data are more normally distributed comparing to the CPM normalized data. So I would highly recommend you to do the log normalization comparing to CPM, and it's also a default normalization method in CRAT and ScanPy also. So, also can be considered as a process of normalization, and it's a standard pre-processing step prior to the principal component analysis, shortened as PCA. So a normal way of doing it is by doing the Z-score transformation. So the normalized count is divided is minused by the mean of that genes and divided by the variance of that genes. So the reason of doing that is to scale the variance of the genes at the same level. So so that when you are doing PCA, those genes with higher expression will not dominate the principal components. So different from the log normalization or other normalization. So the scaling is done for every individual genes at gene level. So after that, what we got is 3 types of matrix. One is raw count, one is normalized, and the other one is the scaled data. So they are used in different places for the downstream analysis. For example, for the highly variable gene the detection, we will use the scale data, and sometimes for some of the functions or methods, they will use the normalized data as the input and calculate the variance and scaling it inside the function. And for dimensionality reduction PCA, you have to use the scaled data. And for DEG analysis, we will usually use the normalized data. And for visualization in heat map and dot plot, well, what we use is the scaled data. And as for like feature plot or Violin plot, we will use normalized data. And sometimes we could also use the raw count data for the visualization of these genes. So after the normalization, we will do the dimensionality reduction. So this is important because because the initial input of matrix of single cell data is of high dimension, usually for one sample, we will see like more than 20,000 genes expressed in the sample, and also with the expected number of cells is usually 10,000 per sample for these technologies. So you will have more cells when you are doing the integrated analysis. So dimensionality reduction is very important because it can project high dimensional data into lower dimension. Here, usually we use 2-dimensional or 3-dimensional. And while it can it have to retain the essential properties. Here is the biological difference of the gene expression. So there were a lot of popular algorithms like PCA, UMAP, TSNE, diffusion maps, and FATE. These three are widely used, and these two I will introduce later as the algorithm for like lineage tracing. So in single-cell RNA sequencing data analysis, dimensionality reduction is not a one-step process, and it goes from very high to find highly variable genes, and to PCA, and to UMAP. And the first step is to find highly variable genes. So the hypothesis is that most of the genes are not differentially expressed. Only a very small part of the genes are of very high variance. So you can look at the plot. You can see that these are genes with high standard variance are of very small parts, and a large part of it are of very low SD. We will only use these highly variable genes as the input of the PCA analysis. Here, usually we choose 20 highly variable genes according to the variance. Sometimes we will use more, like 3,000 in some of the work. So after that, we will do principal component analysis. So principal component analysis, which is PCA, is a widely used in linear dimensionality reduction method. And so it can project data into new subclasses that can explain the most variance of the data. Later, we will use the results from PCA as the input to do the clustering, and as well as the second round dimensionality reduction. So the reason why we are not using PCA for itself is because of the large number of cells. The top two principal components are not good enough to show the heterogeneity of the cells in single cell. So here is the example. So this plot is the results, PCA results from bulk RNA-seq, and the number of samples are limited. And so you can see that the samples are well separated according to the stimulation state and the stimulation time. And the cell type of stimulated samples are separated with each other. It's good enough. However, in single cell RNA sequencing here, we can see that some of the clusters are very close to each other, and it's not well embedded into the 2D space of the PCA. And therefore, we need to use more PCAs here for the second round dimensionality reduction, like UMAP and TSNE. And here is a question of that: how many PCAs should we use for like downstream analysis? So basically what we do is to plot a alpha plot here like this, according to the SD of each of the PCAs, and we choose the top PCAs until it's getting flat. So here is the 10, but sometimes if you want to choose the optimal PCA number automatically, here I would suggest you to use some of the toolkits. Like for example, here is a tool called Find PCA, developed in R, and it has inbuilt many of the methods to automatically choose the PCA number. For example, here is the suggested one called perpendicular line, and it will draw an alpha plot for the top 15 PCAs and draw a perpendicular line here from 1 to 50, and to calculate the distance to the perpendicular line for every PC. The maximum distance from the to the perpendicular line will be set as the number of PCA you should use for the downstream analysis. Here is also 10. I would suggest you to look at these types of tools, and it's helpful. And also in CERAT, there is a procedure called gestural procedure, and it will calculate the significance of every PC here, drawn. So after this procedure, I'd give you some suggestions to how many PCs you shall use for downstream analysis. So after PCA, what we'll do is to do the secondary nonlinear dimensional reduction. So the most frequently used algorithm is UMAP and t-SNE. So here we can see that the PCA is not good enough. PCA results are not good enough, but in the UMAP 2D space, the cells are well located. So these are cells, the clusters from different cell types are located very separately from each other. And however, comparing to t-SNE, so UMAP is computationally faster, especially if we're dealing with cells for very large amount of cells, like 1 million cells. So that in that case, t-SNE may take a couple of days for the computation. So I would suggest you to use UMAP in that case. And also, UMAP better preserves the global structure of the cells. So for example, here, in t-SNE space, we will see that two parts of the CD8 positive cell, T cells, are separated by the CD4 positive T cells. But in UMAP, these cells are well enabled to each other, and it's located besides the CD4 T cells, and it's more reasonable. And besides for the t-SNE and the UMAP, there are some other algorithms designed for lineage tracing. So these types of algorithms, like the diffusion maps and FATE, captures more continuous change of gene expression. So it can be used to learn cell differentiation or cell state transition. And this type of dimensionality reduction results seems to be more like a trajectory, and it is more useful in that case. So PCA is not only used for dimensionality reduction, it is also used for KNN clustering. So what we use now for single cell clustering is the graph-based clustering. So it takes the results from the PCAs, and it will calculate the distance measures for every pair of cells. And then, based on the distance matrix, we will build the KNN graph, which is K nearest neighbor graph, as well as the SNN graph. And after that, when you give a resolution to it, the cells can be clustered. Like here is the illustration of the graph-based clustering. So the graph is a KN graph. So here, every node is every cell, and the weighted edge is shows how strongly the two cells are connected to each other. And by setting a resolution, like for example 0.3, we will cut the edges that is lower than 0.3. Those badly connected cells will be separated with each other, and you will get two clusters. So if you want to get more clusters, you can set a higher resolution. So one question is how to resolution. Sometimes we see people using 0.1, and sometimes we see people using a very high resolution, like 1.5 or something for subtyping analysis. So these are hard to say whether it's good or not. So here I would suggest some of the tools that are designed to evaluate the resolutions, like For example, here is a tool called class tree, and it will build a clustering tree for a sequence of resolutions from very low to high, and you will see how the clusters are changed from lower resolution to higher resolution. Like for example, here these clusters in point 1 will be separated to 2 clusters at a 0.2 resolution. And based on this, you can have some suggestions for which resolution you shall choose. And also you can visualize the gene expression of some of the marker genes on this clustering tree to see whether you can cluster that cell type at which resolutions, and this also help you to choose the resolution. On the other hand, there is another software called MR3, and it kind of evaluates the ARI score of different number of clusters K to evaluate the performance of or the stability of the clustering. And we shall choose the one that is when the plot is getting very flat and stable. So the next part, when we have these clusters, what we do is to do the marker gene detection. So marker gene detection is actually the differential expression analysis, which is DE analysis, and it is designed to find significantly differentially expressed genes or DEGs. So if it's for bulk RNA-seq, we can only do the analysis for groups or samples under different conditions. But for single cell, because we have different clusters, so we can compare clusters to get these cell type specific genes, and these are have a high chance to be the marker genes. So there are a lot of the analysis tools available, and some of these are designed for bulk RNA-seq, like HR, DE-seq2, Lima, and so on. And these can also be used in single cell RNA sequencing. However, these type of softwares have very limited computational speed, so maybe try to use a DE analysis that is built in Seurat or ScanPy. Like for example, here is the example of how we do DE analysis in Seurat. And after we have got these clusters, like for example, we want to find the cell type or cluster specific genes of this cluster, which is cluster 3. We will set this one as the target group of cells, which is ident1, and setting the rest of the cells as the ident2 or the background cells. So by comparing the two groups of cells, we will get these types of table. So the average log FC, which is the log fold change of the average expression between the group. So if it's higher, means that it's more upregulated in the target group, which is cluster 3. And so for PCT.1 and PCT.2. So PCT.1 is the percentage of the cells express that gene in the group in ident1. So the PCT.2 is a percentage of the cells expressed in ident2, which is the background. So the marker genes are those genes with very high log fold change and high PCT.1, but very low PCT.2, and which you can see that it is only expressed or highly expressed in this target group. So here, for example, class 3 detected the genes of MS4A1 and CD69A. These two genes are the marker gene of B cells, and so that we can know that this class 3 shall be B cells. So as for the statistical analysis, so there are a lot of statistical tests, and here I show you the evaluation of different differential expression analysis tested in a published work, and from the all these factors evaluated, we see that Wilcoxon ranked sum test will be showed a overall very good performance, and we choose this one for like this default DE analysis for single cell RNA-seq, because the advantage of using Wilcoxon ranked sum test is that it is a non-parametric test. So no matter how the gene is gene expressional distribution is, it can use it. However, if we are using t-test, we have to be assured that the gene expression is normally distributed. So the next part is to do the cell annotation. So based on the unsupervised clusters, here like we have 9 clusters, and the gene expressional pattern, which are the DEGs we detected from the analysis, we can annotate the cell clusters. The first way of doing the cell annotation is by annotating cells manually. So by looking at the table, and as well as the so the exact gene expressional pattern through the violin plot or feature plot, we can annotate cells like for this cluster, we think it is the B cell. And the question is how we can know which genes are cell markers. And so there are 2 ways. One is from the published papers. So for every single cell published paper, we shall see some descriptions like this to telling you that how they did the annotation and which genes they used for annotate different cell types. So by using these genes, you can do the annotation by yourself for your own data. So the other way is from the databases. So here I listed some of the databases we widely use. One is CellMark, one is PanZonedB, and one is Signecosis, that is developed by Singleron. So these databases have collected as many so many genes, marker genes from different cell types from different tissues, and both all of those have the marker genes for human and mouse, and for you to, so you can check the marker gene from these any of these databases. So the another way to annotate cells by using some softwares to annotate cells automatically. There are actually a lot of annotation, automatical annotation softwares. Here I listed 2 of those. One is Singleron and one is CellID. So Singleron is designed to annotate cells from previously annotated reference data sets. So if you have a data set of the same cell, same tissue origin, and you can annotate your own query data by comparing to the reference data set and calculating the similarities, and you will the software Singleron will predict the labels for you. So like this. So Cell ID is another one, and it's published last year, and it is a little bit different, and it annotates cells from a list of cell type signatures. So these cell type signatures come from the databases or your own collection. So you can make your own list of marker gene signatures, and you the software will calculate the enrichment cell score of different gene signatures for different cell types and for different cells. And after that, the cell ID will predict the cells for you. So here is the example. So the left one shows the actual cell type, and the right one shows the prediction from cell ID. You can see that a big part of the cells are correctly predicted. However, still some of the cells, you can see that in gray, these cells are unassigned to any of the none of the cell types. And so this is to say that so far none of the software can do the cell prediction annotation automatically with very perfect correct rate. So no matter what software you use, you have to check manually according to the predicted results and do some correction to it. So so the cell ID automatic annotation is also included in the Synacosis databases, so you can try to do it automatically on the database. So the last part is the enrichment analysis. So after we got the list of genes that are differentially expressed, and you can do the enrichment analysis. So this is very similar to bulk RNA-seq. All the methods of bulk RNA-seq for the enrichment analysis can be used for single-cell RNA-seq data. Like, for example, to use the pathways from GOKGD or like reactome, you can do these types of enrichment analysis. And also you can do PPI or GSEA, GSVA. It's very similar to bulk RNA-seq. I will not go through in detail. So something special for enrichment analysis in single-cell RNA-seq is that you can do cell scoring of functional gene sets. So these gene sets can be the pathways from the listed databases like GOKGG or others, and it can also be the cell type signatures from single-cell RNA-seq, like from. So this concept is very similar to cell ID. So you calculate the enrichment score of different cell type signatures, and it will predict the cell type for every single cell. So there are a lot of methods designed for cell scoring, like AU-cell, U-cell, add module score in CIRAT, and SSGS-EA. And the concordance of these methods are all very high, so you can choose one of those. So here I would like to introduce two of those methods. One is AU-cell. So AU-cell is kind to ordering the genes in a cell according to the expressional level. So these are the rank of the cells, and by calculating the area on the curve inside, whether the genes are inside the gene sets or not, you will get a enrichment score. So for every cell, you get a enrichment score, and so that you can visualize it on the t-SNE or UMAP plot to see that which cells have higher activity of that gene set and of that function. So it's very useful. So like U-cell is another one, so very similar to AU-cell. It can also like score every cell for the functional gene sets. However, it is comparing to AU-cell, the computational running time of U-cell is extremely low. So it's a very high efficiency. So maybe you can also use U-cell as a choice for cell scoring. So that's all for my talk for today. So thank you for our team and as well as all the audience today for this talk. So here, if you have more questions, you can visit our website or email us as well. So here, let's go to the Q&A.
Yes, thank you so much for the very interesting talk. For differential gene expression, using different methods, will the resulted genes be very different?
So it can be different. So because the statistical value can be different. So the log FC, PCT1 and PCT2 should be exactly the same. However, when you are using different statistical tests, the p-value and adjusted p-value could be different. Those genes like this with very different gene expressional pattern should be identified no matter what statistical analysis you use, but sometimes those are of less changes in different groups that could be different.
Can you elaborate a bit on using reference single-cell RNA data, for example, Brain Atlas, and how to integrate using them in single R or other approaches, especially with respect to normalized or scaled data?
Sorry, I actually didn't get the question. Could you say again?
I can read it again for you. Can you elaborate a bit on using reference single-cell RNA data, for example, Brain Atlas, and how to integrate using them in single R or other approaches, especially with respect to normalized or scaled data?
So actually, in single R,
I'm sorry, just to add some more, what are the major pitfalls when using external sources like from the Brain Atlas?
So it's like for a single R, it actually imported some of the data sets from, like, for example, the mouse brain atlas or human cell atlas. So these types of reference data can be used for annotating the cells. And for and there are also when integrating different data sets or from different technologies, we have to deal with batch effect. So they are kind of so those cells are treated and captured in different technology and from different labs or considerations. So these can be very different, and the overlaps of the expressed genes can be very different with each other. So when you are integrating these cells, these data sets together for use with your own data, the battery effect shall be considered. And I think later in webinar 5, we'll have more details about how we could integrate data and how to dealing with these types of questions.
For more information, visit our website at www.singleron.bio, or for any question, just write us an email. Thank you so much and have a great day.
Thank you so much.
Thank you. Bye-bye.
Bye.